Good afternoon, everybody. Welcome to this ERKNet ESPN webinar on the genetics of human renal agenesis and renal dysplasia. My name is Tom Nijnhuis. Before introducing our speaker, I would like to introduce myself. I'm an adult nephrologist from the Radboud University Medical Center in Nijmegen. So I just said I don't have that much experience with this topic, so it will be very interesting for me also today. I'll be your moderator today. But of course, I would like to introduce our honorable speaker for today, which is Professor Adrian Woolf. Professor Woolf is a professor and chair of pediatric science at the University of Manchester in the United Kingdom. He's also an honorary consultant nephrologist at the Royal Manchester Children's Hospital and an honorary professor at the University College in London. He has, I think, over 25 years of experience as a clinician scientist, where he wants to pinpoint genetic and environmental causes of congenital malformations, the topic of today, define roles of implicated molecules in organogenesis and differentiation, and design and test novel therapies preventing these malformations and promoting regeneration. So his expertise actually spans from, I think, molecular physiology via genetics to clinical nephrology. And therefore he also runs a renal genetics clinic in the Royal Manchester Children's Hospital, but also a similar renal genetic clinic for adults with kidney problems in St. Mary's Hospital. And today he will give a lecture on the genetics of human renal agenesis and renal dysplasia. Professor Woolf, the floor is yours.
Okay, thank you very much. Can I just do a final check? Can you hear me and see the screen okay? Yeah.
For me, everything is okay. Yeah.
Okay. Okay. Thank you very much for the introduction. As you've heard, I work in Manchester, UK. That's in the north of England. And half my time I work in the university, where I try and do laboratory studies to work out why people are born with abnormal kidneys and ureters and bladders. And the other half of the time I work in the Children's Hospital, where as you heard, I've been running a renal genetic clinic now actually for over 10 years, and that's a multidisciplinary clinic. So there's myself as a pediatric doctor, we have a clinical geneticist sit in with us, and we also have an adult nephrologist sit in with us as well, so that we can assess the whole family. So the overview of my talk today is as follows. First of all, I'm going to start off by telling you some clinical aspects about human renal malformations. And I think many of you will know this already, but maybe there'll be some adult nephrologists here who are a little bit less familiar with this clinical background. After that, I'm going to give you a genetic example of this thing we call renal agenesis, where the kidneys are not there at all. And then in the rest of the talk, I'm going to give you two examples of what we call renal dysplasia, which is when the kidney begins to grow, but it becomes full of abnormal, poorly functioning tissue. And the two examples I'm going to give you, one of them will be where the problem is intrinsic to the kidney itself, and that causes the dysplasia. And the second example I'll give you is one in which initially the kidney is actually quite happy, but it becomes dysplastic as a secondary effect to lower urinary tract obstruction. So this is the clinical background. Well, all pediatric nephrologists know that about half of the kids with end-stage kidney failure were born with malformed kidneys. And if you do the mathematics and work that out worldwide, probably between 20 and 90,000 children are alive today on dialysis or having had a transplant who were born with malformed kidneys. But you know, even more important than that, in some ways, numerically certainly, is that about 1 in 5 young adults with end-stage kidney failure were thought to be also born with malformed kidneys. And in the last couple of decades, there's increasing evidence that such individuals carry mutations of genes that drive the growth of the kidney and the lower urinary tract before birth. And in this picture here, we can see a sliced open kidneys showing you some of the different phenotypes. So in the big box on the left, we have a sagittal section through a normal kidney. The most mild type of malformation is called a hypoplastic kidney, where the kidney is still got glomeruli and nephrons in it, but it's smaller than normal. I'm not going to talk about that today. What we will talk about a lot is this other entity called the dysplastic kidney, and you can see some pictures of these on the right-hand side of the box. They can be smaller than normal or bigger than normal, and they often contain cysts. And the other entity that we'll talk about today is called renal agenesis. And I can't draw a picture of that for you because there is no kidney at all, and usually the ureter is absent as well. So there was a very great histopathologist who worked in Chicago in the USA in the last century called Edith Potter. And she did some amazing microdissection studies of normal human embryonic kidneys and also fetuses that have been terminated and were found to have dysplastic kidneys. And in these very careful microdissection studies, she described on the left the normal recurrent branching of what we call the ureteric bud, and this is the remnant that will branch to form the collecting duct tree. In contrast, when she did microdissections on dysplastic kidneys, she found that this branching phenomenon was much impaired. There were very few normal nephrons, and many of these branches terminated in cysts. And this is different to what we would call polycystic kidney disease, because in polycystic kidney disease, the early development of the kidney is essentially normal, and the cysts only form later on in the preformed tubules and nephrons. Whereas in dysplastic cystic kidneys, the cyst formation is an innate part of the abnormal development. If we would look at a dysplastic kidney down a microscope, if we had some histology, it would look like this picture on the right. There would be some simple tubules surrounded by stromal tissue. And this picture would be maintained throughout life. And in fact, this picture looks rather superficially like a very early normal embryonic kidney. And on the left-hand side of the screen, I've got histology here from a 6-week gestation human kidney. And you can see it's also very simple with a few tubules and some stromal tissue. But in the dysplastic kidney, it never really gets much beyond this stage. And therefore, many dysplastic kidneys either produce no urine at all or very little urine. In recent years, we've realized that some of these entities, like renal dysplasia, can be inherited. And just to make the point, this is one family that I used to know when I worked in London. And you can see 3 generations are affected by kidney dysplasia. In the final generation, 2 of these individuals have died because they had such severe renal dysplasia, they died very soon after birth. But there are other individuals in the family who didn't have such severe renal dysplasia, and some of them could start a family of their own. And in generation 2 and in generation 3, I've indicated 2 individuals by red arrowheads. And very interestingly, these individuals were misdiagnosed when they presented with kidney failure in adulthood. The man was thought to have focal segmental glomerulosclerosis, but in fact, he was born with an absent kidney on one side and a dysplastic one on the other. And in generation 3, an individual was found to have chronic kidney disease and a little bit of proteinuria, and she was misdiagnosed as having minimal change nephrotic syndrome. Again, that was wrong. She had bilateral kidney dysplasia. So let's go into the main part of the talk now, and I give you some examples. Let's start with this thing called renal agenesis, where the kidney never forms at all. This can be unilateral, and if the other kidney is okay, you can lead a relatively normal life. But if you have bilateral or both kidneys with renal agenesis, you obviously are in serious trouble. Well, you are only in serious trouble after you're born, because that's when you start to need your own excretory function. And it's interesting to know that a fetus that has bilateral renal agenesis can survive all the way through gestation, because of course the waste products that build up in the fetus cross across the placenta and then appear ultimately in the pregnant mother's urine. So the genetic example I want to give you here is called Fraser syndrome, and it's not to be confused with Frazier syndrome, that is completely different and is a genetic disease of the glomerulus. Fraser syndrome is caused by a mutation of a gene called FRAS1. So let's look at this a little bit more. These individuals not only can have unilateral or bilateral renal agenesis, but they have other clues. They have what we call a multi-organ syndrome. And the clues that people with Fraser syndrome have are that they're born with membranes over their eyes. This is called cryptophthalmos, meaning hidden eyes. And they're also born with short and fused fingers and toes. Now, we know that mice can get exactly the same sort of Fraser syndrome as humans can get. And shown here on the left is an embryonic mouse with Fraser syndrome. And you can see it's got these dark hemorrhages over its eyes and at the tips of its lower limbs. And these hemorrhages are actually due to dissociation of the dermis and the epidermis. And when these heal up, you get scarring, and that's what causes the cryptophthalmos and the fused fingers and toes. But more interestingly to us are the pictures of the renal tracts. So the middle frame here shows a late gestation renal tract from a mouse with Fraser syndrome, and you can see 2 normal adrenal glands, you can see 2 gonads and a bladder, but there are no ureters and no kidneys. And in contrast, the frame on the right shows a normal litter mate. This is a non-mutant mouse, but in the same litter. This mouse has got 2 normal kidneys, and they each have a ureter. So the reason why these mice, and we believe also the humans with Fraser syndrome, get renal agenesis is as follows. In normal development, the kidney begins to form when a little branch of the Wolffian duct, or the pronephric duct, branches from the distal end of the pronephric duct, and this is called the ureteric bud. And this ureteric bud will grow into cells called renal mesenchyme next to it. And it's the interaction of these 2 components that will eventually form the branching collecting ducts and the nephrons. And the problem in Fraser syndrome is that the ureteric bud either never initiates, or if it starts to grow, it never reaches the kidney mesenchyme. And the result of this is shown here. If you would look in these mutant mice a couple of days later and try and find the kidney, you would find a tiny structure shown by histology on the right. And this structure is actually the kidney mesenchyme that is dying by fulminant apoptosis or programmed cell death. And it's dying because it normally is rescued by the ureteric bud. In contrast, if you would look at a normal littermate, a non-mutant littermate kidney, you would see the picture on the left. You would begin to see some tubules and early nephrons. So Fraser syndrome is usually caused by mutation of a gene called FRAS1. It's actually one of a member of several Fraser syndrome genes. I won't talk about the others, but they're all thought to work in the same way. And what this gene does, it encodes a gigantic protein. It's about 4,000 amino acids long. And this protein coats the surface of the ureteric bud in normal development. And we think you need this protein coating the surface of the ureteric bud to mediate growth factor signaling between the mesenchyme and the bud. So we don't exactly understand how, but without this protein, the signaling does not occur and the kidney never really begins to grow. And just lastly, coming back to this idea of renal agenesis, it's very useful to have the mouse model of Fraser syndrome. And we found that because growth factor signaling seemed to be the key problem here, we were able to rescue the development of the kidneys by breeding the Fraser syndrome mice with another mouse strain that would upregulate growth factor signaling. So if you just, I'll take you through this because it's a kind of a remarkable thing, really. So on the top left is the renal tract of an embryo, a normal mouse embryo showing 2 normal kidneys and 2 ureters. The middle picture is similar to the one I've shown you already. It's a Fraser syndrome mouse embryo. There are no kidneys and no ureters. But when we cross this mutant mouse with another mouse, it's called the sprouty mouse, that upregulates growth factor signaling, instead of having no kidneys, we suddenly have a mouse that still has the Fraser syndrome mutation, but it grows 2 kidneys. And this is shown on the right. And these mice are actually born and they live a relatively normal lifespan. They still have a membrane across their eyes and they still have fused fingers and toes, but they have 2 kidneys, albeit a bit smaller than normal, and the kidneys work reasonably well. But when you look at these kidneys down the microscope, you also see something quite interesting. You see that many of the glomeruli have glomerulosclerosis. And in fact, this Fraser syndrome gene is not only necessary for the initiation of the kidney in very early embryogenesis, but later on, it's also necessary as a matrix protein to maintain the viability of normal glomeruli. Right, let me now give you 2 genetic examples of renal dysplasia. And remember I told you that what renal dysplasia means is that the kidney begins to grow in the embryo and the fetus, but it doesn't really differentiate properly. So if you look inside that kidney, you'd see lots of undifferentiated tubules and few, if any, normal glomeruli. So the first example I want to show you, and many of you I think would have seen some of these individuals, are individuals with mutations of HNF1B. This stands for hepatocyte nuclear factor 1B. And this disease was only really described about 20 years ago. So when I was at medical school, this disease was not recognized. But in pediatric nephrology, we now believe that mutations of this gene, HNF1B, are the commonest defined cause of a child being born with renal dysplasia. And the renal dysplasia in this context is part of a multi-organ syndrome. And you sometimes hear this referred to as the renal cyst and diabetes syndrome. Sometimes you see this running in a family from one generation to the next in a dominant manner, but in other families, you just see one individual. And in those individuals, there's usually a de novo mutation of this gene. In fact, this gene was initially associated with one type of diabetes called MODY number 5. This is maturity onset diabetes of the young, number 5. And when HNF1 beta mutations were first described in those individuals, it wasn't really realized that they could also get kidney disease. And then it was noticed that some of them did get kidney disease, but it wasn't the classic diabetic nephropathy. It was they had dysplastic kidneys, often with some cysts. And this is a little example of a girl who I used to look after when I worked at Great Ormond Street in London. And it just shows you the spectrum of problems that these individuals can get. Before she was born, the routine antenatal scan showed that she had a right multicystic dysplastic kidney. And as often occurs, this spontaneously involuted in the first couple of years after birth. So she was left with a solitary functioning kidney. Now, if you or I had a solitary functioning kidney, we would hope that it would be bigger than normal, because if you have a solitary kidney, it should take over the role of the absent kidney on the other side. But with this girl, we noticed that her solitary kidney was not enlarged, and that showed that it was also diseased. And indeed, it was echo bright on ultrasound scan. At 12 years of age, she developed overt diabetes mellitus. And the reason that some of these children and teenagers get this is that you also need HNF1B to form your pancreatic islets. And then finally, to add insult to injury, she had a third type of complication of this syndrome. When her periods were due to start later on, when she was 13, she presented with acute abdominal pain. And this was due to retained blood in a malformed uterus. And if you're a female, you also need this gene for normal uterus development. Now, if we went back to her antenatal scan, it would have looked something like the picture on the left, where you have a normal kidney on one side, in this case the left kidney, but the right side is this enlarged kidney with cysts. This is what we call a multicystic dysplastic kidney. And then after birth, if you really wanted to investigate this very thoroughly, although we tend not to do this nowadays, if you would do, say, a DMSA isotope scan, you would find that this multicystic kidney had no uptake at all. And as I said about this individual, and as often occurs with children with multicystic kidneys, the multicystic kidney can be big at birth, and then it spontaneously involutes. So these are two ultrasound pictures of a child we looked after in London. The picture on the left is an ultrasound soon after the child was born. You see an echo bright kidney with these very big cysts. And then a couple of years later, you do another scan, and it's almost impossible to detect the kidney. That massive kidney has spontaneously involuted to just a tiny little fragment. Now, how do mutations of HNF1B cause renal dysplasia? Well, in order to understand this, we ought to try and understand where HNF1B is normally expressed. So if you look at a normal human embryonic kidney, this is shown on the left, and look for the RNA, which in this picture appears purple, you find it's highly expressed in these branching collecting ducts and also in some forming nephrons. So it's very widely expressed in all epithelia of the developing kidney. And if you look at the protein level by immunohistochemistry, you would get a picture as shown on the right. And this gene codes for what we call a transcription factor, which is a nuclear protein. And as you would expect, on the immunohistochemistry, you see these brown nuclei of the tubules. And HNF1b works as a transcription factor to switch on and off a whole array of other genes. Another interesting thing about these individuals is that if they survive and you follow them through to the teenage years or the young adult years, you find that they often have electrolyte abnormalities. Particularly, they often have a low plasma magnesium. And this is because the HNF1 beta gene is not only necessary for the formation of tubules, but after the kidney tubules are formed, you need HNF1b to maintain physiological functions. And we think the magnesium wasting may be explained because normally HNF1b transactivates a gene called FXYD2, which is implicated in magnesium handling in the distal convoluted tubule. Now, just to finish off the HNF story, I'll show you a couple of other slides going into the more futuristic research realm. What we're trying to do is to understand this disease using pluripotent stem cell and kidney organoid technology. So we start off with patients that we see in our renal genetic clinics. And here on the left is an MRI scan of one such teenager. And you can see his small cystic dysplastic kidneys. He was in fact about to start dialysis. And he donated a blood sample to us, and we take the white cells from there and make pluripotent stem cells from his white blood cells. And then what we do is we ask if we differentiate these pluripotent stem cells towards kidney organoids, will they form normal kidney organoids or will they look different to normal kidney organoids? And further down the line, if they do look abnormal, can we understand the molecular things that are going wrong in these kidney organoids? And the long-term idea here is to find druggable targets so that one day we might be able to give these individuals a drug that would make their kidney differentiate more normally. So in this picture here, I'm showing you on the left a normal kidney organoid. Actually, this was made by cells from an unaffected mother. And on the right, I'm showing you a kidney organoid made from mutant HNF mutant cells that we made from the blood cells of the patient. And you can see the mutant kidney organoid is very different from the normal one. It has fewer structures, and the structures are bigger than normal. And in brief, if you would look at the histology of the normal organoid, you'd see the picture on the left with some glomeruli and some normal color tubules. But the HNF1 beta mutant organoid is full of very abnormal structures. For example, these multi-layered tubules that are bigger than normal. And as I said, we're looking for druggable targets to try and move the dysplastic phenotype to a more normal phenotype. And we're doing this initially by looking at the very broad array of genes expressed in the mutant kidney and then compare it with the normal kidney. And this would be the topic of a different discussion, but one thing that is very notable is in the mutant kidney, there is a downregulation of many proximal tubule genes that are involved in tubule physiology. For instance, LRP encodes for megalin, very downregulated, AGT, angiotensinogen, and so on. This seems to recall what I told you earlier, that these individuals, as they go through life, can develop a tubulopathy, where they waste magnesium and other ions. Lastly here, I want to say that HNF1B can be inherited in a dominant manner, and you may see other families that you think of HNF1B inherited in the same way, but they may in fact have mutations of other genes like PAX2, EYA1, GATA3, and indeed UMOD and MUC1. So these entities can all mimic HNF1 beta related disease. Okay, the last few slides I want to show you is another example of renal dysplasia, but here the problem is not intrinsic to the kidney, but the kidney becomes dysplastic, it's injured as an innocent bystander due to lower urinary tract obstruction. And the example I want to give you is caused by mutations of myocardium. And myocardium, like HNF1B, is another transcription factor. But this transcription factor is essential not for the formation of epithelial tubules, but it's essential for the formation of smooth muscle in the bladder and in the ureter. And if you have a myocardium mutation, you form a bladder, but there is no smooth muscle or no useful smooth muscle. And you end up with a phenotype that we call the prune belly syndrome. You have a massively distended bladder that fails to empty normally, and urine cannot drain from the kidneys, and the back pressure cause obstruction to urine flow and secondary dysplasia of the kidney. Most of these individuals are detected before birth, and here are pictures of a fetus that's been terminated on the right with a massively distended abdomen due to the massively distended bladder. And the other pictures are of MRI scans of an unborn fetus with prune belly, where you can also see the massively distended bladder. And usually these individuals are terminated, and we tend not to see many of them born anymore. But I remember when I started out in pediatric nephrology a couple of decades ago, we did see these individuals not uncommonly. And in an international collaboration a few years ago, we published 4 families that were unlucky enough to have babies with prune belly syndrome, and they all carried mutations of myocardium. And one interesting thing about the genetics of this myocardium mutation is that you only get the severe bladder disease if you are a male carrying the mutation. If you're a female carrying the mutation, you only get mild bladder disease, but later on in life, you get heart disease. And you get heart disease because myocardium is also needed for formation of some of the cardiac valves. We don't really understand why males should be more affected in this way, but we suspect it's because males have longer urethras, and the normal bladder of the male needs to generate a bigger pressure in order for that bladder to empty. And this is a very rare thing that I've never seen before, but we were fortunate enough to be able to look at the histology of one of the terminated fetuses. And the bladder is shown in the upper right picture here. This is a mid-gestation terminated fetal bladder with a myocardium mutation. And in the wall of the bladder, you can find some muscle fibers that they're very ragged and vacuolated. And if you look at this individual's kidney, shown in the lower right, you can see some tubules and glomeruli, but the glomeruli are cystic. And this cystic blowout or dilatation of the Bowman space is very, very typical of renal dysplasia secondary to lower urinary tract obstruction. And just for comparison, on the upper left and the lower left, we have a time matched normal fetal bladder, showing the normal muscle bundles, and the normal kidney, showing the non-cystic glomeruli. And this is a scheme of what would be happening in one of these prune belly renal tracts, shown on the right. We'd have a massively dilated bladder, we call it a megabladder. This would then cause obstruction of urine flow. You'd get secondary hydronephrosis, and these glomerular cysts that on ultrasound scan characteristically appear as a row of small peripheral cysts. And just to finish off this talk now, we're aware now that there are several other genes that can go wrong to give you abnormal bladders and secondary renal dysplasia. For instance, HPSC2 and LRIG2, when they're mutated, they cause the Ochoa or urofacial syndrome, and CRUM3 mutations cause a mutation of the acetylcholine receptor on detrusor muscle, and that also results in prune belly syndrome. And there are other genes as well that I don't have time to talk about. So this is really summarizing what I've tried to tell you. I've given you a genetic example of renal agenesis, the FRAS1 gene, and I've given you two genetic examples of human renal dysplasia, one caused by HNF1 beta mutation, where the developmental problem is intrinsic to abnormal kidney tubule maturation. And I also gave you a second example of renal dysplasia, where the dysplasia is secondary to a problem in the lower renal tract, in this case caused by myocardium mutation, causing megabladder. I just want to thank everyone I work with in Manchester, the geneticists, my other clinicians, stem cell biologists, and thank all of our funders. And I leave you with this slide of the future meetings of ERKNet. And I'd be happy to answer any questions that you have.
Okay, thank you very much, Adrian, for this very nice overview of this topic. And I guess you could have filled the whole evening or maybe 2 days on this topic, but at least we have a good overview on what we should think of when encountering these problems. I already see a lot of questions coming into the question box here, but if people would still like to ask questions, please put it in the question box, and then I can ask them to Adrian. And actually, the first question that came in was the question whether there is also a risk of malignancy in the dysplastic kidneys, which would also mean that you would have to follow them with echography or another type of.
That's a very interesting question. And when I started out in pediatric nephrology, we, and I think many other departments, if they saw a child born with a unilateral multicystic dysplastic kidney, they would remove it surgically. And the reason for that was there was a story that some of them could turn malignant in later years, and a second story that they could cause severe hypertension. But the epidemiologists tell us that the risk of cancer is no greater than, having a normal kidney, and also the risk of the multicystic kidney causing hypertension by itself is also debated. So we no longer take out these multicystic kidneys, and we wait for them to spontaneously involute. But it's an interesting question.
Another question coming into the question box is, do you routinely perform genetic studies in children with unilateral MCDK? That, of course, also raises another question. How do you actually or what is actually your diagnostic workup once you see that there is a problem like this?
Yes, so that's a good question about whether to do gene tests in someone with unilateral multicystic dysplastic kidney. So the way I approach this in our renal genetic clinic is as follows. First of all, I have a close look at the opposite kidney. Now, if the opposite kidney is hypertrophied, as we expect, I have slightly less interest in doing genetic tests. But if the opposite kidney is also dysplastic, I consider that a more serious situation. That child may end up with end-stage kidney disease over the years. So that alerts me clinically that this is something more important. Of course, we take a careful family history, and if we would find any first-degree relatives with dysplastic kidney, then I would be more alerted to doing genetic tests. And finally, we're looking at these children not just at the kidney and urinary tract, but we're looking at the whole child. So, do they have syndromic features outside the renal tract that would lead us to a genetic diagnosis? For example, someone with a cystic dysplastic kidney and diabetes mellitus, I would immediately think of HNF1 beta. Or someone with a multicystic dysplastic kidney who is deaf, I would think of EYA1 mutations. So I think I don't have a blanket policy. I try and assess the whole patient and look at the whole family, and then yes, we would do a genetic test.
Okay. Another question coming into the question box is, can these patients with kidney dysplasia develop glomerular disease? And I guess you already showed us that there could be glomerular injury in the Fraser mice, right?
Yeah, yes. So this is
but I guess this question is a little bit more general.
Yeah, well, this is a very interesting question. And as I showed you in the example of Fraser syndrome, that same gene that is needed for the initiation of the kidney is needed for the maintenance of glomeruli. And this may be one reason why in some of these children born with dysplastic kidneys, as they grow up, they then develop progressive renal failure. Another reason for that could be that you're probably aware of this hyperfiltration hypothesis that Barry Brenner made several decades ago. So he would say that if anyone is born with a kidney with fewer glomeruli than normal, they will grow bigger than normal, they will hyperfilter, and then they can become prone just from physical forces within the glomerulus to get glomerulosclerosis. And the third cause could be that in someone born with a dysplastic kidney, you may still have an element of obstruction of urine flow that you haven't corrected. And if that obstruction to urine flow, for example, from a bladder that doesn't empty fully, that can continue to damage the nephrons and the glomeruli as you grow up.
Okay. The thing that I was wondering is, well, I'm always intrigued about these HNF1 beta patients because the phenotype can be so diverse. And even within families, the phenotype can be different. Do you have any ideas on what makes, let's say, the phenotypic difference? So what is actually the cause? And I was wondering, can you see that back in the organoids? Because I always think, well, these are patients with the same mutation or deletion. So I would think in the organoids you would end up with the same result. And that would tell you something about whether it's something genetic or it's actually something acquired that makes the phenotype differ.
Well, this is a great question, and I just answer it in two parts. You know, one family we look after in Manchester with HNF is a great example of what you're saying. There's a father who carries the mutation, and he just has a few renal cysts, and he also has acute gout, which is part of the syndrome. And he passed on the mutant gene to 3 children. One of the children was so severely affected with bilateral renal dysplasia and oligohydramnios that that individual underwent termination as a fetus. And then there are 2 other brothers, and they had an intermediate severity. They were born with cystic dysplastic kidneys, but they still retained some renal function. And actually, as they grew up, they've developed tubulopathy as well. So, you know, in one family, you have this whole spectrum of severity. Now, I believe probably this is due to the presence of modifying genes or polymorphisms of modifying genes, but I can't prove that to you. But when we make organoids from these patients, so far we've elected only to make organoids from individuals who are born with quite severe dysplastic kidneys. Because when we study the organoids, we want to try and model that developmental disease in a dish. I can't really tell you what an organoid would look like if we would make the cells from an individual with HNF mutation who's got, you know, relatively normal looking kidneys. Maybe then the organoid would also look relatively normal. But it's, you know, these are very interesting questions that I can't fully answer.
Okay, so still a lot to be
Yeah,
to be found out, I guess.
Yeah, and it of course makes genetic counselling quite challenging.
Yeah.
Because when the families say, you know, if we have another child, will they be severely affected or not? We have to, you know, give them a very open answer, really. The other thing I would say is my genetic friends tell me that maybe there's no such thing as a dominant disease. And, you know, if you look at other so-called dominant diseases, for example, tuberous sclerosis, or maybe even to an extent autosomal dominant polycystic kidney disease, there can be a lot of variation even within a single family. So it doesn't only affect this HNF1 beta disease.
Yes, I agree. Another question from the question box is, are there also non-genetic causes of renal agenesis or dysplasia that we should actively look for? For instance, medications used by the mother, certain toxic substances?
Well, that's a great question, and I believe that there will be environmental influences that interact with genetic influences that cause the dysplasia. But I can give you an extreme example. For example, mothers who, when they're pregnant, have very poorly controlled diabetes mellitus, and this is nothing to do with HNF1 beta gene mutation, they can give birth to babies that have something called a caudal regression syndrome. The lower half of the body fails to develop properly, and this can include renal agenesis. So that's a definite example of teratogenic effect causing renal agenesis. There's also some thought that in mothers who ingest a very large amount of alcohol, the kidney can also be at risk there in the so-called fetal alcohol syndrome.
Okay. Well, looking at the clock, we are already approaching 5 o'clock already. And I see that there are no more questions in the question box remaining. So I would like to thank you, Adrian, again for this very nice talk. And I would like to ask the audience again to take note of our next webinars, which have been at your screen now already for about 15 to 20 minutes. So you will probably have seen them.
Longer than my talk.
Yeah, that means that the discussion was very lively, which is a good thing. So the next
actually, all the questions were really great questions. You know, those are all exactly the right questions to ask. Unfortunately, we don't know all the answers yet.
Yeah. So I would like to also thank all the attendees to being here. Adrian again for being here, and I hope to see you again at one of the next ERKNet webinars. Thanks again. Have a nice evening.
Bye.